Thank you for joining me on UCSD Guestbook. Tonight it is my pleasure to welcome UCSD Professor Theodore Bullock, a highly distinguished neuroscientist and one of the founders of the neuroscience community here in La Jolla. Welcome, Ted.
Thank you.
Ted's an inspiring figure who has had an enormous influence on the way research is done in the field of neuroscience. There are few aspects of the structure and function of the brain that you have not investigated. Ted combines an insatiable curiosity about almost all subjects with an imagination and creativity to devise decisive experiments that illuminate the issue at hand. He's often focused on very large problems, such as, for example, the way in which neurons give rise to behavior, and has searched for the general principles that underlie this process. Ted, let me start by asking you about some of the early work that you began in the 1940s, studying a variety of nervous systems in invertebrates. At that time, you carried out some elegant studies of the conduction of electrical signals, action potentials in the nervous system, also looking at the connections, the synapses between nerve cells. What first drew you to these problems?
Because the state of the science at that stage had shown that there must be synapses, but it wasn't clear yet whether they're general or whether they were an achievement of evolution that only came later on. And it was my good friend Adrian Horridge who showed in the very early 40s that jellyfish have nerve impulses, have this miracle of all or none spike-like nerve impulses that we had wondered before about the evolution, whether they were an achievement of higher animals. He showed that they were already present in these very first or earliest or simplest nervous systems. And I was interested. I had done my thesis on nerve nets in a very primitive animal related to us, the most simple animal related to us, hemichordates. They have, in fact, a nerve net, which was a surprise to me, something like that of a jellyfish. And therefore we found that evidence of synapses there and in the squid giant, where there was a particularly favorable case that will lend itself.
So these electrical signals, both the action potentials that are propagated and the synaptic potentials that transmit the information from one nerve to another, these would seem to be fairly universal principles for all nervous systems.
Yes, and so now the question which really fascinates, intrigues me, is what has been, in fact, the main achievement of evolution, given that nerve impulses and synapses and many transmitters and modulators are very general. And yet the evolution from the simplest nervous systems to that of the most complex animals, like ourselves, is the biggest gap that I can span, I should say, of complexity that I can imagine in the universe. And what has happened, in fact, in evolution is one of the main questions that intrigues me.
Yes. Well, you have written and thought deeply about this issue of what distinguishes a complex brain from a simple brain, either in its structure or its function. How are you thinking about that now?
Well, I've addressed the question of whether it could be just numbers of nerve cells and numbers of synapses, basically size of the brain, and decided no, that cannot be the main thing, although size and numbers of nerve cells certainly is a factor that may help. But it still escapes me, and we are still struggling to find out what are the steps, what I would call emergence, that have explained these several, not many, but several major saltations or increases in complexity that have happened between jellyfish and worms, worms and bees and octopus and lower fish from higher mammals.
Yes.
There's some major changes in the central nervous system that we still have to identify.
Yes. In that connection, I know that you've long been fascinated by the process of information coding in the nervous system by the electrical signals that are generated. Today, as you know better than I, many investigators focus on rate coding, the rate at which electrical signals are produced, or they focus on spike timing, the timing of these electrical signals. But of course, as you have pointed out, there are many other codes. And one naturally asks, why are there so many different codes in the nervous system? And for example, do different kinds of information get coded in different ways?
I suppose so. I don't know the answer to the why question. It's like asking why did any change happen in evolution, but evolution has somehow arrived at or brought into being these diverse simultaneous codes in a system that is not like any computer that we know, but like an extraordinarily highly parallel computer with many things happening at once at the same time. And even in the same nerve cell, it may be encoding different messages at the same time in different ways, with not only nerve impulse all on spikes, but with slow events, with chemical, with electrical events, perhaps even mechanical changes in the shape of the contacts.
So these parallel streams of information then that we take in and process are perhaps coded in many different ways, it would seem. You've also done a lot of research on brain waves, popularly called, an example of which would be the electroencephalogram, the EEG, and you've carried out these studies on a wonderful range of vertebrates and invertebrates, porpoises, dolphins at the vertebrate level, octopus, if I remember correctly, among the invertebrates. Here you're focusing on a different level of organization of the nervous system and really perhaps looking at cognitive functions. What have you learned from some of these studies?
That's a very open-ended question. We used to call it a $64 question before inflation. Expressions like good question are so ambiguous, aren't they? Well, I think that what attracted me to them is simply that they were being neglected, that nobody was, or very few people were studying actually the phenomenology of the field potentials, the summated activity of populations or organized assemblies better of neurons, and that they should be studied as a biological phenomenon. How have they evolved and what could they tell us, even though it may be true that most of it is like the noise of your car. It may not be of actual value as signals to other parts of the nervous system, but in fact I believe some of it may be actually causes rather than just effects. But even if they were just effects, I think we should study them as biological phenomena. And it has amazed me from early days that the spontaneous ongoing electrical activity of, for example, crayfish and virtually all the invertebrates that we studied, worms and snails and insects, that are very different. Their field activity, the compound activity that you might call brain waves, is very spiky with very diminutive or inconspicuous slow waves, whereas our own, which is the same as the mice and elephants and porpoises and sharks and rays and all the vertebrates, have mainly slow waves, and spikes can be had only by searching for them and having special microelectrodes. What could be this difference? I don't know what the meaning of it is, but there's something there that means something about the population structure. Maybe it's synchrony, maybe it's the cooperativity of the population, and the phenomenology is still waiting to be adequately described.
Would this be perhaps one of the distinctions between less complex and more complex brains, this difference in the population activity of the nerve cells?
Partly, I think so, but it's an equal mystery. How come the brainwaves are so similar between frogs and humans, between sharks, rays, and fish, and mammals? From mouse to porpoise, they look essentially the same, and yet I think that the difference in complexity of the brain must be real and tremendous, in fact, between simpler fish and highest mammals, and we haven't found the electrical correlate. I'm sure that there is some feature of the EEG in humans and monkeys and cats that will differ from each other and will differ from the EEG of a frog or a fish, but we haven't yet found those features, and it's the thing that drives me right now. What are the descriptors that we're missing?
Yes, yes. You're touching here in your remarks on the power, I think, of comparative studies, the power of studying the same processes in a wide range of different animals. And you have argued that differences are as important as commonalities in some of your writings. What are perhaps some of the additional benefits that come from this comparative approach to neuroscience?
Of course, mainly you're satisfying your curiosity. What in the world has evolution achieved when you face the fact that what evolution mainly does is to create differences? It creates new species, and not only new species, but new genera, and new families, and new phyla, new types of organization, and we may know very superficially what is the coelom like and what is the skeleton like that defines these major taxa or groups of animals, but we don't know much about what are the differences in their brains. And it's very hard to get the people who are the main experts on the function, that is to say behavior, to agree that this animal is more complex than that. I'm sure personally that the behavior is much more complex in some animals than in others, although I can't cite good authority for saying so, and I think that must come from the structure as well as the physiology of the population of nerve cells, and it seems to me the biggest reservoir of new principles to be discovered, and therefore just intriguing for finding out major principles.
Well, I think you put your finger on something very important there. I think the opportunity for uncovering really new modes of operation of the nervous system is out there for perhaps the next generations of neuroscientists to come along and explore, following in your footsteps. In your career, Ted, you've trained an extraordinary number of graduate students and postdoctoral fellows. My count identifies, I think, 34 graduate students, some 120 plus postdoctoral fellows. This is, by any account, a phenomenal training record. And I want to ask how you have managed to accomplish this. The list of these people reads like a who's who in neurosciences. These folks have gone on to become almost as distinguished, some of them, as you are yourself. What are your secrets of training so many people so well?
I enjoyed it, and I both invited some of them, and some of them invited themselves. The students have just been, by luck, wonderful students. Postdocs have been a great joy. I mean, they're half the fun, besides uncovering secrets from the wet globs of protoplasm. They were the fun of working together, and I've just been lucky.
Yes, yes. Well, let me make a further observation that perhaps has contributed to this. Throughout your career, you have encouraged many of your colleagues, really an extraordinary number of your colleagues, to publish their work independently. And you've refrained from putting your name on their work, as many scientists, of course, do today. And I wondered if there were perhaps a philosophical bent that led you to this practice, or how you came to this process of encouraging the students to take ownership of the work that they had done.
I couldn't say or think that I thought it was a philosophical question, but it just seemed natural that given the diversity of PhD theses with which I was blessed, and I was playing the role of a critic of the suggestions they were making, and making some other suggestions, making alternative suggestions so that they always had a choice. It didn't seem to me the thing to do to claim that I was responsible for this thesis. It was their thesis.
It obviously had it didn't hurt your career in any way, and I think it perhaps inspired the careers of your students and fellows who have, as I mentioned, gone on to wonderful accomplishments following your leadership. Another feature of your wonderful career that stands out is the way in which your research has taken you to various exotic parts of the world to do research on site. You've been to Alaska, to Hawaii, to Japan, to Australia, back to Germany and Yugoslavia, down to Brazil. What have been some of the benefits for you of carrying your research out of your lab down at the Scripps Institute of Oceanography here in La Jolla to these different locations?
Oh, that's hard to say. Certainly, I wanted to show that, as my friend and colleague Per Scholander showed, that you can do some sophisticated physiology by taking your equipment and setting it up in an old schoolhouse or on the deck of something in a remote place. And that way you get access to animals that you can't bring into the laboratory. And I was already well on this path when Scholander applied for and got from the citizens of the United States, fortunately, the ship Alpha Helix, which was designed for this purpose, to take teams of physiologists and biochemists to field locations up the Amazon and into the ice in the Arctic and so on, to look at organisms that they can't get into the laboratory. And that has been sort of a leitmotif that I've enjoyed doing, and quite a task sometimes to entice laboratory scientists who have a big agenda and are busy and happy doing what they're doing to take a month off and see what they can learn with some animals that they had never thought they could get access to in the upper Amazon or somewhere.
Well, it may be beyond the scope of our discussion this evening, but I remember you recounting wonderful tales of doing experiments on the pitching deck of the Alpha Helix or in the depths of the Amazonian jungle with a battery-powered equipment recording from an animal that you just found in the rainforest there. And I think this is a wonderful way to extend the comparative analysis that you've really pioneered in many important ways. Quite recently, you were in Brazil, I think, in Manaus, for the dedication of a building that actually bears your name and that of Walter Heiligenberg, one of your early fellows, at a national institute there. Could you say a little bit about that institute and what it does?
The INPA, the National Institute for Amazonian Research, is a major group of laboratories in various parts of the country in the city of Manaus, in the middle of the Amazon, the state of Amazonia, a city of 1.5 million and 1,000 miles up the river. There are 4 campuses of this institute with 44 buildings and 600 or 800 staff members, many departments, and they just gave money for a new building to a former student who got his degree here, Jose Alves Gomez, who is now has a new building for behavioral physiology, which is exactly the name, or one name for the field that I and Dr. Heiligenberg worked in. And they invited me to come down and cut the ribbon for this new building.
Wonderful.
Which was a great honor and pleasure.
Yes.
Just a few days ago.
Yes, yes. You've been a leader in establishing the field of neuroethology, the field to which you just referred, and this is, I think, a growing area, sort of leading out of neuroscience. What is the future of neuroethology, and does it have a promise of helping us understand the complex behavior of mammals such as human beings?
Well, promise would be a pretty strong word, but I think it has that potential. And It's exactly addressing the question that I've referred to before of how in the world did our brains come to be the way they are? What were the stages by which the brains evolved? Certainly the increase in complexity was not automatic or inevitable or linear, but it happened at the ends of this and that twig of the evolutionary tree. And my concern is that whereas a lot of people are interested in the process of evolution, how it happens, very few are concerned with what actually happened, that is the consequences of evolution. What is different about our brain in terms of the pathways and the connections from that of reptiles or of fish or of invertebrates. We know some of the things, but they're very first order, and I think there must be a lot more. And neuroethology has the promise, especially because it emphasizes comparative studies of different species. Sometimes even closely related species are quite disparate in the way in which they do similar, where they solve the problems of life.
Looking back now, perhaps a bit over your career, what were the big questions that excited you as a young neuroscientist, and how have the big questions that you hope to excite the new crowd of students coming to La Jolla, how have the big questions changed over the course of your career?
Oh my goodness. That's a very tough one too. It would take a lot of homework to answer that, really. Certainly there were questions that motivated, drove, and enticed me, such as whether the synapse, this was in the early 40s, middle 40s, is basically electrical or chemical. And at that time,
fundamental question.
It was quite up in the air. We luckily found that we could record both just before the synapse, just after, just presynaptically, just postsynaptically with intracellular electrodes. In a very, very favorable case, that's one of the advantages of comparative studies. You can find favorable animals in the giant systems of squid, not octopus, not cuttlefish, but in squid. There you can put microelectrodes just on the two sides, and we could show that it could not be electrical. We couldn't provide positive evidence that it has to be chemical, but we could show that it could not be electrical.
Do you think that the big questions in front of us now perhaps focus on the higher levels of organization of the nervous system, some of the complex behavioral processes and issues of cognition in various animals?
Certainly the ones that I'm aware of or that intrigue me, yes. There may be lots of others that I'm not into because I'm just too ignorant, like the chemical bases, and that's where the action is and where lots of great major discoveries come, and magic bullets and useful knowledge. But certainly the origin of complexity, the origin of organizational features that explain our behavior is a big open question that I'm aware of. While I pay obeisance to this great frontier where most of the papers and part of the national programs of national meetings are, namely the biochemical molecular bases.
But I think one does not want to lose sight, Ted, of the importance of the physiological, the functional, and the behavioral studies that really give meaning to the molecular studies to which you allude that are so prominent these days. Our time is drawing toward a close here, but I want to ask one question that I think will be certainly fun to ask. I'm not sure how you will respond to it, but if we imagine for a moment that there were an almighty deity of whom you could ask one question and have that question be answered, given those boundary conditions, what question would you ask?
Expecting him to answer me.
Yes, or her.
Or her.
Yes.
That's certainly very tough, considering that I personally can't believe in an almighty deity. That would be putting too much responsibility for all the stumblings in evolution and inventions of varmints and
Yes.
unhappy
But you know
animals.
where I'm going here. You know
Yes.
I'm interested in what is the big question that's on your mind that you would really love to see solved or addressed, answered.
The reason that's hard is because I'm still at the stage of descriptive natural history. That is to say, I think that the brain has many principles yet to be unraveled, to be uncovered, major principles that will be of very diverse sorts in order to explain this infinitely complex achievement, our brain and the basis of our behavior. So I can't put my finger on one question that says, how does this happen or how does that one happen, that's uniquely important, whereas I'm sure there are a lot of such questions. What is the meaning of synchrony in the brain?
Yes.
What is the meaning of the 40 hertz waves that are so popular these days, and which I have found in lots of behavioral states in lots of animals and lots of parts of the nervous system? It isn't as though they're unique, you see.
Certainly for me, that would be also a very satisfying question to see answered. Well, I wish we could go on further, but this has been a most enjoyable conversation. Thank you very much for joining me here on UCSD Guestbook.